Welcome back. We are starting with chapter 2. And in chapter 2, we will only cover section 2.3, which is the fixation and staining of the microorganisms. The reason being that we have already covered the microscopy in detail in the biology 8420, which is the understanding cellular processes. So we're not going to repeat the same material because it is basically the same. So we are starting with the fixation and staining. So the first thing that what we are going to understand from this chapter, so the staining specimen help to visualize and identify the microbes. So essentially, when a bacteria, archaea, or any of the microbes are present and we need to visualize them under the microscope, we need to stain them. Before staining, we need to fix them. We are going to talk about that, what a fixation process is. And then we're going to plan a series of appropriate staining procedures to describe unknown bacterium as fully as possible, different types of bacteria. And also we are going to compare that how the gram-positive and gram-negative cells during each step, how they differ, and how the two different staining method actually stain them in a different ways. And this is a very, very common process, and we are going to talk about that in detail as well. So essentially, the fixation is done because the specimen which are actually examined by the bright field microscopy are often need to be fixed, stained before examined. The reason for being that they are really microscopic, they are small, and until and unless you actually contrast them against the background, you are not going to see them because they are transparent, they are very, very small, very thin, and they were almost transparent. So that's the reason that we need to actually stain them. So before staining, we need to fix them. So essentially what the fixation process is, the stained cells seen in a microscope should resemble the living cell as closely as possible. So that's the reason the fixation is the process by which the internal and the external structures of the specimens are preserved in position. And it inactivates the enzymes that might disrupt the cell morphology and toughens the cell structure so that they do not change during the staining and the observation, and the microorganism usually is killed and attached firmly on the microscope slide during the fixation, so that when you actually do stain the specimen, they will not be washed off by these several procedures. And there are two fundamentally different types of fixation. One is a heat fixation and a chemical fixation. So in the heat fixation is routinely used to observe the bacteria and the archaea, and typically a film of cells, which is called a smear, is gently heated. So how you make a smear? You actually take a drop of water, and on the drop of water, you just mix with a loop. You just mix a little bit of bacteria and gently actually make a smear. It's a thin film, which is very slightly milkish in color, and you allow it to air dry. It is gently heated, and the heat fixation actually preserves the overall morphology and it inactivates the enzymes, and also it actually destroys the protein in the subcellular structure, which may distort their appearances as well. The important part is that it preserves the overall morphology, but not the internal structure. This is the most important, overall morphology, not the internal structure. On the other hand, the chemical fixation is used to protect the fine cellular substructure as well as morphology. It is used when examining microorganisms. Many electron microscopic technique, chemical fixative penetrate the cells and react with the cellular components and usually proteins and the lipids to render them inactive, insoluble, and immobile. Now, common fixative measures contain ethanol, acetic acid, mercury chloride, formaldehyde, glutaraldehyde. These are used routinely for many different structures, or when we need to look at these structures under the electron microscopes. Now, let's take a look at, after the heat fixation, what are the dyes, and what we are going to talk about it, that how they differ in their different characteristics. So the many different types of dyes used to stain microorganisms have two features in common. First, they have these chromophore groups, chemical moieties with conjugated double bonds that give the dyes a color. And second, they bind cells by ionic, covalent, or hydrophobic bonds. That is the most important part of a dye. And the dyes actually binds the cell by ionic interaction are probably the most commonly used dyes. These ionizable dyes may be divided into two general classes based on the nature of their charge group. Either they are basic dyes or they are acidic dyes. Now, the staining effectiveness of the dyes are maybe altered by the pH, since the nature of the number of the charge moieties a cell component change with the pH, and thus the acidic dyes stain best under the acidic condition, whereas the basic dyes actually do stain best under the basic condition. So what we mean by these, that the basic dyes, so say for example in here, like methylene blue, basic fusion, crystal violet, saffron, malachite green, but they're blue in color. Basic fusion is like raspberry red, violet in color, saffron is red in color, malachite green is green in color, and they have positively charged groups, and they by ionic interactions bind to the negatively charged molecule, such as in the cell could be nucleic acids, many different proteins, surface of the bacterial archaeal cells, and that's the reason that these interaction actually pertain the color to these molecules and make the cells visible. On the other hand, when we are looking at the acidic dyes like eosin, red in color, rose bengal, pinkish in color, acid fusion is also raspberry red in color, and they possess groups such as carboxyl COH, which will be essentially become COA minus when ionized, and the phenolic hydroxyl can become OH minuses as well. And these actually do have these negatively charged groups, and these negatively charged groups actually bind to positively charged cell structure. Now, interestingly, not always we actually do find many of the cellular structures actually don't have positively charged groups. So many times we actually use these acidic dyes to stain the background, and these are used for negative staining. Now, this is a new term that I'm talking about. This has nothing to do with the charge. A negative staining is essentially we are using an acidic dye to stain the glass slide where the organism will appear as clear against a uniformly stained background. And we are going to talk about that in later. So dyes that bind, on the other hand, through covalent bonds or that have certain solubility characteristics are also useful. For instance, DNA can be stained by a fusion procedure in which the staining compound, which is called a Schiff's reagent, is covalently attached to the deoxyribonucleic acid sugars of deoxyribose sugars of the DNA. On the other hand, there is another dye called Sudan 3 or Sudan black, selectively stains lipid. So the Sudan black is lipid soluble. So that's the reason they specifically stain the lipids. The next part that we are going to talk about is about simple staining. So what simple staining is, that microorganisms can be stained by just one single dye. And the simple staining's value lies in its ease of use. The reason for that, the fixed smear is essentially covered with a stain for a short time, and excess amount is washed away. And the basic dyes such as crystal violet, methylene blue, or carbonyl fusion are frequently used to stain and determine the size, shape, and the arrangement of the bacterial and the archaeal cells. So these are some of the reasons that we actually use these dyes. So the next part that what we are going to talk about, the simple stain actually can determine three different things. We can determine the size, we can actually determine the arrangement of the bacteria, and we also actually can look at the shape of the different bacteria. So essentially, simple staining actually gives you three different characters, which is size, shape, and arrangement. Now, the next in the next slide, we are going to talk about another type of staining, which is a differential staining. So the differential staining actually can divide the microorganisms into groups based on their staining properties. And essentially, they or at least more than one type of staining is going to be used in this differential staining. There are many different types, and the classic example is the Gram stain, which is developed in 1884 by a Danish physician, Christian Gram, and is the most widely used staining procedure in bacteriology. So the Gram stain is an example, classic example of differential staining procedure, distinguish organism based on their staining properties. Now, use of the Gram stain classifies most bacteria into two groups, again, gram positive or gram negative. Now, please remember that we have talked about in the previous slides about the different types of dyes. Basic dyes have positive charges. The acid dyes or acidic dyes have negative charges. Now, these dyes actually don't have anything to do with gram positive or gram negative. And we are going to explain that, what we mean by this gram positive and gram negative, nothing to do with the charge. There are also differential stain that used to stain some of the cells which are called acid fast, mean that these cells do have a property that when they are washed with acidic alcohol, they retain their staining. Just as we use that, there are some clothes which are color fast. That means that washing or will not take the color away, color fast. The same way, acid fast means that when you actually use these cells, after the staining process, when you actually wash these cells with acidic alcohol, you will actually retain the stain of the acid fast cells. Most others will be discolored. On the other hand, you can actually visualize the capsules using the differential stains. You can look at the flagellas using the differential stains, and we're going to talk about that in a moment. So the first thing, or the most common one of the differential staining, is the Gram staining, most widely used process. Now, in the Gram staining procedure, in the first step, what we do, that we actually make a smear and stain with a primary stain, which is the crystal violet. So the crystal violet is the primary stain, and we actually just use this. We just make a smear, we heat fix that bacteria, and we actually just put the bacteria there for 1 minute, water rinse, and the cells actually stain purple. In the next step, what we actually do, that we actually use iodine, which is a mordant. Mordant is an helper, and we actually keep that mordant on the slide for about 1 minute, and again we water rinse. The cells actually will remain purple. All the cells will remain purple. Now, in the next, we rinse off the iodine, and in the third step, what we do, that we actually have 95% ethanol, and we call this as an alcohol decolorizer, and we actually destain. Again, the word is destain. We take the stain away by the alcohol decolorizer. We actually expose the cells with dripping alcohol for about 10 to 30 seconds. And after that 10 to 30 seconds, we water rinse. Now what happens? The gram positive cells, here the most important part, the gram positive cells remain purple. That means the alcohol decolorizer will not take the stain away from gram positive cells. Whereas there could be another group of cells which actually initially were stained purple, because all cells remain purple. They were initially purple, but after the alcohol decolorizer treatment, they will now become colorless. So they have now exposed all their dye binding sites, and now you actually add the safranin, which is a pinkish red color, and for about 30 to 60 seconds, you expose the cells to them, and you water rinse and blot dye. So after drying, you're going to visualize them under the microscope. What you're going to visualize, that the gram positive cells remain purple, whereas now the gram negative cells appear red. So what is the principle of that? So the first dye, which is the crystal violet, the cells which will keep the crystal violet or the first stain after decolorizing with the alcohol, is known as the gram positive. So the cells, again repeating, the cells retaining the first dye will be considered as gram positive. And the cells which will be discolored or decolorized or destained by the alcohol application and then they take the second stain as pinkish or reddish color, they will be considered as gram negative. So this actually divides the bacteria in two large groups, gram positive and gram negative, and the property is completely dependent on the cell wall structure of these bacteria. So we are going to talk about that in detail in the next chapter, but here we can just understand that the bacteria can be broadly divided into these two groups, and probably the most important of all differential stain is the gram staining procedure. Gram positive and gram negative are the two different cells that we actually group them into. The next part, we are going to talk about the acid-fast staining. In the acid-fast staining is another important differential staining procedure and can be used to identify Mycobacterium genus. Mycobacterium tuberculosis is actually, or Mycobacterium leprae. These are actually deadly organisms. Mycobacterium tuberculosis cause TB, tuberculosis. Mycobacterium leprae actually called leprosy. And these particular bacteria actually do have very high lipid content in their cell wall, and they contain a particular type of lipid which is known as the mycolic acid. These are a group of branch chain hydroxy fatty acid, and these are really one of the main components of their lipidy cell wall. And this actually prevents the dyes from readily binding to the cell. So to do that, we have to do a specific procedure. The commonly used procedure is known as the Ziehl-Neelsen method, Ziehl-Neelsen. Ziehl-Neelsen method, you will see that it is written in your book. This uses a high concentration of phenol and the carbonyl fusion, as well as a wetting agent. A wetting agent, what we mean by this, is actually reduces the surface tension in between the lipidy cell wall and the dye. Usually, wetting agent used is called tergitol, Tergitol, tergitol. This is a wetting agent, but along with the phenol and the carbonyl fusion, we use this tergitol. So this actually drives the stain or the carbonyl fusion into the mycobacterial cells. So without this, it is impossible for any dye to penetrate this high lipid structure. There are also some other older procedures where you can actually heat the cells in presence of these carbonyl fusion, and that will actually make the cell wall a little bit pliable, or it will allow the dye to penetrate. So that was a heating method. But we usually do not use this heating method anymore. We just use a wetting agent along with the phenol, which actually make the cell wall slightly soft to take up the stain. Once this dye has penetrated, the cells are not easily decolorized by acidified alcohol. We actually use 0.3 molar hydrochloric acid in alcohol, and thus they are called acid fast. So after the staining them, we actually wash the cells, which are colorized now by the carbonyl fusion, which is a raspberry red in color. We actually wash them with the acid alcohol, and the acid-fast cells, which are the Mycobacterium genus, will actually keep the stain and will not be decolorized. If there are any other type of bacteria in there which actually doesn't have any mycolic acid in their cell wall, the dye will actually wash off by the acid alcohol decolorizer. Now what happens that we actually counter stained them with another dye. Usually we used like a methylene blue, which is blue in color. So if in the mixture you have a cell which is essentially non-acid fast, they will color in a different way. So if you just go to the previous picture, you will be able to see right here that you can see that in the acid-fast stain, you have the acid-fast bacteria that you are looking red cells, whereas the blue cells were actually decolorized. They will take up the stain first. They will take up the red stain, but they will be decolorized by the acid alcohol, and then they will actually essentially pick up the second stain, which is now methylene blue, or they will appear bluish in color. The similar way that we can also stain other different structures. So let's take a look. Say, for example, we can actually use the negative staining process as well. So the negative staining procedure is essentially what we call as a capsule stain. So capsule stain is a technique that reveals the presence of a capsule, a network of usually polysaccharides that surrounds the many bacteria and the fungi. And we're going to talk about these capsules in the next chapter in much detail. So the cells are actually mixed with India ink or nigrosin dye and spread out on a thin film slide. After air drying, the cells appears as bodies surrounded by a halo of a capsule in the midst of a blue-black background, because the ink and the dye particles cannot penetrate either cell or its capsule. Thus, the capsule staining is example of a negative staining. There's a little distortion of the cell shape, and the cell can become counter-stained for even greater visibility. So let's take a look at the picture, and we will just again repeat that what we have just said. Say, for an example, we are looking at this picture right here. So the background is looking like, although it's looking like bluish, but it is essentially blackish, essentially. It's just a little bit pseudo colored in the process of the picture. So the background is stained by an acidic dye. So I'm going slow here. The background is a glass slide which is stained by an acidic dye, which is essentially has negative charges. The glass slide is inherently positively charged. So the acidic dye, which has negative charges, is attracted to the glass slide, and thus dye binds to the glass slide. So the glass slide becomes this uniform blackish color, blackish gray color, the uniform color. This is what is called a negative stain. The process of differential staining, we can use another dye, which is a basic dye or a positively charged dye. Again, remember, a basic dye on the positively charged dye, and that dye, addition of that dye, will actually stain the bacterial structure right here inside the capsule. So the rod-shaped structure that you are seeing inside the capsule is the bacteria stained by the basic dye, positively charged, and it is actually attaching to all the negative structures that the bacteria and most of the cells actually do contain. Remember that we already talked about most of the proteins are negatively charged, the nucleic acids and most of the are negatively charged. So most of these cells are negatively charged, which is stained by the basic dye having a positive charge. This particular negative stain, one thing you have to remember that why the polysaccharide capsule that you're seeing in white is colorless when you are using an acidic dye to stain the background, which is the glass slide, or you are using the basic dye to stain the bacteria. The reason being that the polysaccharide is actually neutral. They are not charged. It's polysaccharides is neutral in charge, so they are not positively charged, not negatively charged, so that they are not attracted by the positive dye or the basic dye. And that's the reason that they remain colorless, whereas the bacteria being totally negatively charged, the surface is completely negatively charged, is being stained by the basic dye, and the glass slide being positively charged is stained by the acidic dye, which is negatively charged. So that's basically the principle of the negative differential staining. Is the flagella staining, and this provides the taxonomically valuable information about the presence or the distribution of the flagella on a bacteria and archaeal cells. What we mean by that, these are the flagellas are actually locomotory organs and fine thread-like organelles, and they can only be seen using electron microscope, although bundles of flagella sometimes can be visualized by a dark field microscopy. But on the other hand, what happens that to visualize these flagella under the bright field microscopy, we actually can increase the thickness by coating them with mordants or helpers, such as tannic acid or potassium alum. So we just use the tannic acid or potassium alum, and then staining with a dye such as basic fuchsin will allow the visualization of these cells, or these flagella. So that's what it is being shown here. So you have a special type of stain used for the flagella, as well as a special type of stain is used for the bacterial cells itself. So these are your different types of dyes or the differential staining that we have used. And that's pretty much all that we have used, all the techniques that we need to learn in this chapter. And when we actually go into the next chapter, in the chapter 3, it will make much more sense that where is the flagella, how their distributions actually give them different types of names. Capsule stain, what are the capsules? We are going to talk about that as well. And then the take-home message, what we have, there are many different types of microscopy, and these are basically you have learned in a different chapter, but essentially that the specimen preparation and different staining methods are just as crucial of microbiology for the understanding of this procedure. So that's pretty much all the chapter, and we will wrap up.